June is an immunotherapy month, and I'm sitting here with Dr. Kim Margolin, my colleague and partner at St. John's Cancer Institute. And Kim, you've been involved with immunotherapy research and treatments for quite a long time. At the time when it was considered perhaps dead end of oncology, and it required a lot of faith and belief and perseverance to really get us into the point where we are right now. Give us your perspective on some of the recent developments. Your area, of course, right now is particularly cutaneous malignancies, melanoma. What are we doing here at St. John's that you consider innovative in that field?
Thank you, Przemek. I know we're both in fields that are touched heavily by immunotherapy and have been for a few decades now, but really the last decade or decade and a half has been the real explosion of success finally. Having been in the melanoma field since the beginning of my career, and prior to that, always interested in immunotherapy, I've been lucky enough to see what's happened in melanoma as well as in renal cancer, which you can talk about. But starting out in a field where just about all patients succumb to the disease because the same chemotherapies that are given to many other solid tumors are almost completely without value in melanoma. Right. So it was crying out for something more effective and maybe less toxic. Fortunately, it's been known for many years, mostly in animal models, that melanoma had a unique sensitivity to immunomodulation. And so it was one of the first tumors, along with renal cancer back in the late 80s, that was addressed with immunotherapy. In those days, it was interferon, and then we went to interleukin, which was first T-cell growth factor, was being used to grow what turned out to be the AIDS virus, HIV, and then it became clear that with recombinant purified interleukin-2, you could sometimes see responses of advanced melanoma and advanced renal cancer with nothing more than just that one agent, but it had to be given at very high doses, which were very toxic. So the therapeutic index, so-called toxicity versus beneficial outcome, was still very unfavorable. Over the succeeding years, many attempts to make interleukin-2 less toxic, more effective, better when blended with other immunologic molecules was of very modest success until the immune checkpoint blocking antibodies came along with Jim Allison and his work in observing and Gordon Freeman and people who discovered this ligand receptor interaction between PD-1, programmed death receptor on T cells, and PD-L1, or programmed death ligand, on tumor cells, as well as on many other types of cells that control the overstimulation of T cells in normal life, which we want to keep at bay. And so those drugs turned out to be very, very effective and of much more modest toxicity, particularly the PD-1 and PD-L1 drugs. Wow. And we've been building on that ever since. I'll let you talk for a bit, and then we can come back to what I've been doing.
Yeah, so I think obviously, as you mentioned, the checkpoint inhibitors really transformed the whole field of melanoma and cutaneous oncology. I'm primarily involved in geo-cancers. We really form very heterogeneous group of diseases. So they are kind of anatomically lumped into GU group, but really very different histologies, very different implications for the immune response and so on. And as you know, the major component of GU oncology is prostate cancer, and that has not been that greatly affected by immunotherapy advances. So we have Provenge, which is a kind of vaccine type of approach that has modest benefit applicable maybe to a small number of patients. There's tremendous effort obviously going on in terms of research to try to make prostate cancer more relevant in the field of immunotherapy. At St. John's, we are doing several, I think, really interesting trials. Just a couple of examples with this bispecific antibody in collaboration with Regeneron company that on the one hand targets the very ubiquitous molecule on the surface of prostate cancer called PSMA, and on the other, it attracts CD28 protein on the surface of T cells. So try to infiltrate prostate cancer tumor with T cells, and then also checkpoint inhibitor is added to the mix. So I think that's one of the relatively, I think, innovative concepts in prostate cancer. Another example would be, again, the recurrent theme of the PSMA molecule, along the lines of perhaps antibody drug conjugate concepts, when you have a humanized antibody against, again, this PSMA, very important molecule on the surface of prostate cancer, and you attach radioactive isotope lutetium-177. So we are collaborating with Australian company that's developing that in prostate cancer. So we do a lot of those, I think, innovative trials here at St. John's. And of course, as you know, in kidney cancer and bladder cancer, the checkpoint inhibition became really standard pretty much in the even frontline settings right now. So how about some interesting new trials that you are involved in here at St. John's?
Yeah, so as you alluded to, or as was implied by what you just were saying, what we've really seen is a little bit of a brute force approach where just about every new and interesting molecule is being combined, or new approach is being combined with the blockade of either PD-1 or PD-L1, which is probably sensible, although it may be that, you know, the preclinical folks could do a little better job of understanding exactly how we do that and what's the best combination. But interestingly, in melanoma, some folks are going back to the drawing board with some of the cytokines, and now engineering is going on, molecular engineering is going on therapeutically, both in the cytokines such as interleukin-2, which I talked about at the beginning, and those that are related to it, the so-called gamma-C cytokines. So for example, IL-7 and IL-15 and IL-21, none of those are in the clinic quite yet, but interestingly, we just had the first approval of IL-15 in bladder cancer, which you know about. Sometimes in order to get those cytokines to be safe and effective, or to use them regionally, such as is the case with bladder cancer, you have to really engineer them. So you have to put them together in weird forms that are not found in nature in order to either enhance their activity or to mitigate their toxicities. And all of that is sort of a work in progress. We also see engineering being applied now to these immune checkpoint blocking antibodies. So of course we've had drugs, the PD-1 antibodies such as nivolumab or Opdivo and pembrolizumab or Keytruda for a number of years now, a decade or so, over a decade since their approvals. But now attempts are being made to alter the section of those antibodies that binds to other cells of the immune system in order to either kill the target cell or to otherwise make them do more functions than they otherwise would do. So we're working with one company called Agenus that has been engineering the CTLA-4 blocking antibody to make it do other functions, spread its functions. Other companies are almost all the big pharma and small biotech companies have their own version of PD-1 blockade that they're combining with other things, such as the blockade of one of the newer checkpoints that's called LAG-3, and those combinations are heavily in the clinic, including Regeneron, including BMS, and probably other companies as well.
We have one of those trials.
So we are studying. We actually have 3 trials. They happen to all be with Regeneron, not for any particular reason, but we have an adjuvant trial, so post-operative management of patients with high-risk melanoma who had their primary melanoma removed. If it was either high risk due to the thickness or ulceration or the presence of involved lymph nodes, patients are being randomized among 3 groups. One is the standard pembrolizumab, and the other 2 include a different PD-1 molecule, it's almost the same as pembro, together with one of 2 doses of an anti-LAG. The same general approach is being taken to neoadjuvant therapy now, treatment we give to bulky disease, usually in lymph nodes of melanoma, prior to surgery. That often causes reduction in the size of tumors and making them more amenable to surgery. And the third one is comparing in advanced disease to comparing directly Regeneron's 2 molecules to BMS's 2 molecules, relatlimab plus nivolumab, which is already commercially available as Opdualat. There are vaccines. You talked about the Dendrion agent, which is a form of, a professional antigen presenting cell, the dendritic cell, together with an immunomodulatory product, GM-CSF, attached to PAP in prostate cancer. And we will have a molecule that is also bispecific from Immunocore, which has given us tebentafusp in uveal melanoma from the eye, which is far more rare. In this case, unlike your PSMA, which should be present on all prostate cancer, we have a molecule called GP100 in the case of tebentafusp, or PRAME in the case of melanoma, where those molecules are presented by the transplantation antigens, and they have to be unique to that particular protein. And so only half of the patients with melanoma will have the proper tissue transplantation antigen, the HLA or MHC, to present the PRAME. So that will be only available for half the patients. And the other end of that, yours was CD28 with the PSMA, ours is CD3, but those 2 molecules are very tightly related to each other and stimulating T cells. That's pretty much it as far as what we have, other than what's coming up is tumor infiltrating lymphocytes, but we can come back to that.
So I may add also that along the lines of what you mentioned with this engineered molecules, cytokine, interleukin-2, we have a clinical trial that actually I am PI, although you've accrued most of the patients. In melanoma, the engineered IL-2 that is bound to albumin, and also I think there are genetic modifications that make it less toxic and perhaps activating less these regulatory suppressors to say even more selectively cytotoxic T cells. So that's one example. And what we have here at St. John's is capability to do those kind of trials that are quite involved, including inpatient monitoring, which is, I think, quite unique for non-academic place. We do have infrastructure and bandwidth to do that. I think that some of the cellular therapies hopefully will be coming up. Tell me also a little bit about some of I know that you've been involved also in this imaging clinical trial that I think potentially is applicable in maybe in the future, predicting response to immunotherapy early on, or really identifying patients better for selection for various agents. I think that's very intriguing.
I'll just tell you about that quickly. We are working closely with a biotech company called ImaginAb that one of our former colleagues from City of Hope actually created. Mm-hmm. And basically what it is a shortened antibody. It's a fragment of an antibody called a minibody that lacks the FC portion and therefore is not binding to antigen-presenting cells, but instead it's carrying an isotope, zirconium-89, which has favorable PET scanning properties and a favorable half-life of about 3 days, and that is bound to the FAB, or the antigen binding section times 2 of a normal antibody molecule, and in this case it's against the CD8 determinant on CD8 cells. So the idea is it's a PET tracer for CD8 cells, and the goal of this is to be able to create a technology that allows you to essentially do PET scans on patients, which rather than detecting metabolically active tumors or sites of infection or inflammation the way the FDG does, instead it's detecting CD8s. The idea being that if a tumor is filled with CD8 cells at the beginning of therapy, we know from many experiences that it's more likely to respond to the kinds of immunotherapy we're currently doing. The second best, I think it's safe to say, is that if the tumor is relatively cold and doesn't have a lot of CD8 cells in it, but if you give a therapy that's capable of causing the influx of CD8 cells into the tumor, that might itself be a good predictor for a response to therapy. And then, of course, the least would be cold to cold, in other words, low CD8s to low CD8s. There's internal built-in controls, fortunately, which is that certain organs at baseline, normal organs, have a lot of CD8 cells in them, including the spleen and the liver and the bone marrow. So you can sort of internally test your agent for for whether it's performing the way it's supposed to on the same kind of scan. And because of other properties, we're generally normalizing the signal, the SUV, it's the same measurement, to the aorta, where there's sort of a blood pool. So the interesting that you use the term predict, because there are one protocol that's completed and it's going to be submitted for publication very soon, which was a correlational study of the histology, so everyone had to have a biopsy with the signal on the PET scan, whereas the second trial that is now about 2/3 to 3/4 accrued is called iPredict, and it doesn't require biopsies, but it requires the correlation of the outcome of immunotherapy with the appearance of those CD8 scans, pre-treatment and on treatment. So we'll see how that goes. It could be very exciting, and there's a number of pharmas very interested in using it together with their drugs.
So Kim, over the next 2-3 years, what do you think is the most exciting area of development in immunotherapy that hopefully our patients here at St. John's will benefit from? I know it's tough to predict the future, but I'm giving you a relatively short perspective.
Yeah, Przemek, I think that, it costs nothing to say what we think is going to happen. I'm keeping my eyes on all forms of cell therapy. And of course, that initially became available and very exciting in blood malignancies such as leukemia and lymphoma. It's been harder to develop in solid tumors, but we now have something related. It's not these transgenic T cells, but it's T cells that are unmanipulated from melanoma patients that are taken from tumors, are cultivated outside the patient and given back to the patient, who's prepared with certain chemotherapies that can essentially lead to an up expression of growth factors for the lymphocytes that are given back. All of that, and incorporating interleukin-2. So that's one form of therapy that does not use the checkpoint molecules, although there will be protocols with that in the future. But I think it's important for those patients who couldn't continue with checkpoint blocking because they got toxicities, in a sense, an overabundant immune reaction. So keeping an eye on that at the same time as keeping an eye on some of these bispecific antibodies and the antibody drug conjugates, other things that are like cell therapies, but they aren't quite, because the fact is the rising tide floats all the boats, and so you could be making big strides on one, but at the same time strides are being made in the other, and you may end up going back to a simpler form of therapy. I think that it's almost time to talk about vaccines. You have your dendritic cell vaccine. We have what looks to be an exciting personalized or patient individualized form of vaccine therapy in melanoma that's looking very good in the adjuvant setting in confirmatory trials now. Whether it's going to be ready for prime time in advanced melanoma remains to be seen, but lots of people are studying various forms of vaccine. I, at the same time would like to know what you think about kidney cancer. That's one of your one of the two that's highly amenable to immunotherapy, but can we cure advanced kidney cancer? Is that, that's kind of the holy grail. We've got a lot of treatments, but what do they actually do?
Well, no, I agree with you, and I think you also have background in kidney cancer going back to years of interferon and interleukin, and it kind of mimicked some of the drug development, I think, in melanoma. But yeah, immunotherapy, particularly checkpoint inhibition, is now really the standard component of frontline therapy. There are several regimens that people debate which is the best. Hard to know, but are we curing kidney cancer? No question, we are extending life in a major way, and I think we are curing some patients, although it's still a small proportion of patients, probably in the single digits or maybe low double digits, but there are patients that now we see from these trials with immunotherapy combinations who are in long-term remission for years now, 5+ years. So I think the odds are hopefully that some of these patients are cured, but still, long way to go. But I would echo what you said about antibody drug conjugates. I really feel that, again, this is maybe a relatively intermediate term perspective, but those are the most exciting to me because the technology is already there. It's already been proven in several malignancies, in bladder cancer particularly, and there are efforts in prostate, kidney cancer. And I think mechanistically, I think it's a matter of finding the right target and biochemistry to make it work. And I think those drugs probably will be highly studied and applicable over the next 2-3 years in many settings.
There are hematologic malignancies, as we know. So there are times when a patient really wants to try something, either they've heard of it already or they don't, but you are aware of it and they come in and you don't have an ordinary trial for them that would consist of multiple institutions participating. The FDA calls it, or politicians call it, permission to try or something like that. But I think more correctly would be expanded access or compassionate use protocols for individual patients. My understanding is that sometimes those are made available to patients when a drug is looking pretty good to the FDA, but it hasn't gone through all the hoops. Have you ever been able to do that for a patient?
Yes, I have. And as you point out, there are various mechanisms for that. Sometimes it may apply to a larger number of patients. A study like that is kind of what you've mentioned, called expanded access, where you have a very promising drug already looking very good in terms of efficacy, but not quite approved yet because of bureaucratic hurdles. And the expectation is that it will be likely approved, and the company allows this manufacturer of that or developer of that compound allows patients to be treated on this expanded access, compassionate type of program. So we've certainly done those, but also slightly different mechanism would be an individual patient expanded access or compassionate use, which I've also had examples like that once or twice. So there are various mechanisms to access some of these really promising drugs that are not quite yet formally approved and commercially available. And we have capacity to do that with our clinical research office and regulatory team to get it here for our patients.
Yeah, I think technically the terms are, now I remember, right to try.
Right to try.
And that we don't really approve of because those are usually brought to us by patients who are asking about things that are pretty fringy.
Right.
Whereas a single, I think what you're referring to is a single patient exclusion or some exemption, yes, where you have to get you have to do for one patient what we normally do for many patients, which is to get regulatory approvals. Sometimes time can be not our friend because it still requires jumping through a lot of hoops. But we do what we need to do when we can for our patients.